Good afternoon, everyone. I'm very delighted to introduce Professor Suvarn Kulkarni, my colleague from chemistry department. As you know, he is one of the recipients of the Best Publication Award for the year 2022. Professor Kulkarni, received his PhD from Pune University. He had a couple of postdoctoral tenures. First at Academia Sinica in Taiwan, and then later at the University of California, Davis. And then he joined our department in 2009. Since then, he has established a very vibrant research program in a unique area called glycochemistry. So he focuses on synthesis of total synthesis of oligosaccharides. That means sugar units linked together, several multiple sugar units linked together. Also synthesis of glycoconjugates, sugar units linked to other biomolecules. And his thrust area is development of sugar-based vaccines for the treatment of various diseases like cancer, for which he won the Best Publication Award. Professor Kulkarni has won several awards and recognitions for his, not only for his research contributions, but for his teaching as well. As you know, he is also the Associate Dean academic program right now. So he is a multifaceted personality, but his research is mainly on sugars. So he is a super sweet person. When you interact with him, you will realize. And today he is going to talk to us about expeditious synthesis of rare sugar-containing bacterial glycoconjugates for vaccine development. Professor Kulkarni.
Thank you, Rishi, for a very humbling introduction and very kind of you. Thanks. So I'm also grateful to IRCC Award Committee for recognizing our work, and also thanks to IRCC for having this program. This is, I take it as a really a great encouragement for people who are doing research, and thanks for selecting our topic for this award. Today I'm going to talk about expeditious synthesis of some sugars which are present only in bacteria and how we can use them for vaccine development. This award is given for 3 publications. The first publication, it came in 2013 from my first student, Madhu Emmadi. He single-handedly contributed to this project. That was in Nature Protocol. And the second publication was in JAX. It was contributed again by another student, Someshwar Rao Sanapala. So Madhu synthesized the D-form sugars which were there, which are present in bacteria. Somesh synthesized L-form sugars. And these methods were not there before in literature. So these are highly cited publications because people are using these methods and saying that, okay, this chemistry works. Then the third publication is again JAX, contributed equally by 2 very talented students, Archana Mai Behra and Diksha Rai, who is in the audience. And so I would like to begin by thanking their efforts. And none of this would have been possible without their continuous efforts and tenacity and intelligence. So carbohydrates, as probably you know, that they are decorated with sugars, just like in tablet. So these sugars are like a first entry of contact, point of contact for bacteria or virus. And they're involved in all these important life processes such as infection. And structurally, they're diverse and complex and microheterogeneous in nature. So if you want to develop any carbohydrate-based drugs, if you try to isolate carbohydrates from natural sources, you will get a microheterogeneous mixture. And you won't be able to do any pharmaceutical chemistry based on that. So that's where chemists like us come in picture. This is again one more cartoon showing you the presence of glycolipids and glycoproteins on this our cell surface. And due to their position, we're well positioned to interact with other cells to carry out a variety of functions such as cell-cell communication and signaling. For many years, it was believed that these sugars are only there in higher organisms such as us. But last 15 years, this notion is disputed. And now what we know is that bacteria also have sugars. For example, this is a TEM image taken in Israel. It shows you that bacteria, these all appendages are nothing but sugars present on the bacteria. And there's a difference though, like for mammalian glycans or glycans which are present in us, we have this set of 10 sugars which makes up our glycome. But bacteria have thousands of sugars. A bacteria can modify these sugars. One of the very peculiar point is that bacteria have these amino sugars. Like many positions are deoxygenated and many are functionalized with amine groups. And due to this, bacteria have certain sugars, this group of sugars, this is a D form, these are L form, just the mirror images of those. They are present only in bacteria and they are absent in human beings, in the host. And that's where our work started in 2011. We started thinking, can we first synthesize these sugars and use them as a specific diagnostic or for vaccine development, because bacteria have those and we don't have. So our system is going to recognize them as foreign particles. So here, quick few structures where you see that these are the colored sugars. These are only present in specific group of pathogenic bacteria, and their absence and presence has been linked to pathogenesis. So we thought that, okay, we need to synthesize these sugars. And when we looked up the literature, we saw that you can do it from sugars or amino acids, but all of them were really, really long, 20 steps processes, and very, very difficult to make. So here, Madhu's role starts, and I discuss once with Madhu, can we do this a certain reaction that if we start with this commercially available sugar and do a double displacement, I will not go into nitty-gritty of organic chemistry, we will reach these bacterial sugars. And I had no idea at that time that this one discussion will keep me busy next 10 years. And indeed, Madhu showed that if you use a bulky nucleophile, you can selectively displace one triplet and then add another triplet to make a set of all bacterial rare sugars. So this work was published in Nature Protocol, and this is very well a process tested around the globe, and people are using this method. So that helped us to synthesize a variety of glycans which are there in pathogenic bacteria. For example, this is from Neisseria meningitidis, Bacillus cereus, Streptococcus pneumonia, and so forth and so on. And these are the deuterionic oligosaccharides which were then subsequently synthesized in our lab. And just before closing, before going for his postdoc, Madhu also completed the synthesis of this hexasaccharide. Then Somesh came on board and we asked these questions, are there L sugars? And we found that there are many L sugars in bacteria, but no method. So we asked, can we extend the same method, just protecting one hydroxyl and doing double displacement in a one-pot manner? And Somesh, to cut the long story short, he was able to show that yes, if you start with commercially available L-rhamnose and L-fucose, you can then get access to all the L sugars. Then we assembled D and L form containing sugars, and at that point of time, Archana Mai Behra and Diksha Rai came on board, and we synthesized some interesting glycans. And at that time in 2017, WHO declared a list of priority pathogens. So that gave another direction to our research. So they listed a group of pathogens which are resistant to antibiotic, and they call it escape pathogens, and they said that WHO declared that there's a need to urgently come up with a new antibiotic or vaccine for these escape pathogens. And we identified 3 pathogens, Staphylococcus aureus, Pseudomonas aeruginosa, and Acinetobacter baumannii. And this was done by Archana and Diksha. Just making these sugars is not enough. We have to put them together, rope them together stereoselectively. For here, example, I'm showing you formation of an alpha or axial bond here between these 2 sugars. And this helped us to make these 2 trisaccharides, and I'm very glad to say that this particular sugar, trisaccharide, is already showing immunological activity and is useful for vaccine development. So we are taking further this, and it has this linker. So this linker is attached for further conjugation to proteins for further immunological studies. So that's where we are, and Diksha Rai recently completed also these 2 complex tetrasaccharides from Acinetobacter baumannii, Now, these Pseudomonas aeruginosa, Staph aureus, and Acinetobacter baumannii, these are dangerous pathogens. They're opportunistic pathogens. So let's say you get admitted in the hospital, and if there's a prolonged admission, in that month or two, you will get these infections because these are hospital-borne infections. And if you get those infections, even vancomycin will not work against them. So you will go for something else, and you will die because of something else. So that's the reason that there's an urgency, because this will lead to multisystemic failure. These pathogens, so they look not so dangerous because they are not like TB or something, but it's really, there's an urgency to find out some drugs or therapeutics or vaccines for this. So now we are focusing all our energy in our lab, only looking at these 6 pathogens and looking at their carbohydrate structures. And we are collaborating with Professor Peter Seeberger at Max Planck, where now we are putting conjugation here. So the idea is you have this trisaccharide, we have the linker that we attach to a conjugate, to a carrier protein, and this conjugate is then first we do a microarray to see whether it is reacting with the serum. And once we find that, then we are going to inject it in mouse and then do the immunological evaluation. So at this stage, I'm very glad to share with you that one of the compounds is active and we are taking it for further vaccine development. So after another 10 years, probably I'll get another ICC award, and then at that point of time, I'll be able to tell you the full story. This is my last slide. So we are here. So we have synthesized the bacterial oligosaccharides. Now, all these, and we have also done this glycotope selection, right? And now we are making the glycoprotein synthesis. So we have the glycan, we are attaching to carrier protein CRM197, and then it will be subjected to formulation. Then this work is remaining. So animal immunization, monitoring of immune response, and then actual, you infect the mouse and then you test your vaccine. So I would say 40% of the work is done, 60% is remaining. So we are looking at another 10 years work going ahead. So with this, what remains for me is to just thank this wonderful group of people I'm privileged to work with. Madhu Emadi, my first student who started all this work. He is in Germany and is working in a vaccine company. Someshwar Sanapala. So Madhu did all the work on D sugars, as I mentioned. Somesh did all the work on L sugars. He is a faculty at IIT Tirupati. Archana Mai Behra, who did the trisaccharide synthesis, which got the activity. She is now a faculty in Utkal University. And Diksha Rai, who just got a PhD and aspiring to go for postdoc in some good lab. And I'm very thankful to all the other students as well for their contribution. Very thankful to Department of Chemistry for support. Thankful to all the funding agencies without which nothing can be done. And I was telling in my class the other day that we can dream of these projects just because we are working in IIT Bombay, where we have great infrastructure and there's a great growth of infrastructure in the last 10 years. So I was fortunate to join in 2009. So I've seen the best days of IIT Bombay, I should say. So you name the instrument, we have it. There's no limitation. So our ideas is only the limit. So I'm very thankful to all the people who worked for that. And of course, thankful to my family and friends for their support. Thank you.